Tardigrades were first discovered in 1773, and since then, scientists have identified approximately 1,200 species, but researchers believe many more are waiting to be found. These tiny invertebrates typically range from about 50 micrometers to just over 2 millimeters in length, making them barely visible to the naked eye. Their body structure is surprisingly organized for something so small. A tardigrade has a cylindrical body divided into 5 distinct segments. The first segment holds the head, while the remaining 4 each carry a pair of legs that gives them 8 legs in total, each tipped with small claws that help them navigate their environment. The two main groups of tardigrades differ in some key ways. Eutardigrades tend to have translucent bodies with flexible, pliable skin. They are primarily found in freshwater environments or on land. Heterotardigrades, on the other hand, often have cuticles reinforced with protective plates. This group includes many marine species that live in the oceans. Despite their miniature size, tardigrades possess remarkably sophisticated internal anatomy. They have a complete digestive system that runs from mouth to anus, allowing them to process food efficiently. Their diet varies widely depending on the species. Some feed on algae and bacteria, while others are predators that hunt even smaller invertebrates. Their nervous system is equally impressive. A brain sits at the front of their body, connected to a series of nerve clusters called ganglia that run along each segment. They also have sensory organs that help them detect their surroundings, including simple eyes and various receptors for touch and chemical signals. Reproduction among tardigrades takes several forms. Some species have separate males and females, while others rely on parthenogenesis, a process where females produce offspring without fertilization. Their eggs often have elaborate protective shells, sometimes decorated with intricate patterns. These tiny creatures continue to surprise researchers with their complexity. Every new study seems to reveal another layer of sophistication in their biology. Tardigrades have managed to colonize an extraordinary range of habitats across our planet. They can be found in freshwater environments, marine settings, and terrestrial ecosystems on every single continent, including the frozen landscapes of Antarctica. This remarkable distribution speaks to their adaptability. As a group, the terrestrial species have particularly interesting living arrangements. They make their homes in mosses, lichens, and soil. But there is a catch to their terrestrial existence. These tiny animals depend entirely on thin films of water that surround their bodies. Without this moisture layer, they cannot survive in their active state. This dependency explains why they are often found in damp mosses and lichens, where moisture tends to linger even when conditions become drier. When it comes to food, tardigrades are surprisingly versatile eaters. Their diet spans a wide range of organic matter. They consume algae and bacteria, feed on fungal cells, and even hunt small invertebrates like rotifers and nematodes. Some species have been known to eat other tardigrades as well. This dietary flexibility likely contributes to their success across so many different environments. The tardigrade life cycle follows a pattern that might sound familiar to anyone who has studied insects or other arthropods. Females lay their eggs either directly into the environment or inside old exuviae, which are the shed cuticles from previous molts. After hatching, young tardigrades go through a series of molts, typically between 2 and 7 times. As they grow, the young already resemble adults in form, but they reach sexual maturity only after their second or third molt, which happens over several days. Once they become adults, tardigrades typically live for several months. One of the most remarkable tricks tardigrades have up their sleeves is something called anhydrobiosis. The word itself tells you what it means. Anhydrobiosis translates roughly to life without water, and that is exactly what happens when conditions become too dry to support normal life. Tardigrades can push the pause button on their own metabolism. They lose almost all the water in their bodies and enter a dormant state that can last for years. When water returns, they simply rehydrate and pick up where they left off for a long time. Scientists knew this happened, but did not fully understand how. A major breakthrough came in 2023 when researchers James Fleming, David Pisani, and Kazuharu Arakawa decided to map out the genetic foundations of this ability. They focused on several tardigrade-specific gene families that produce what are called abundant heat-soluble proteins. These proteins seem to protect cellular structures when water disappears. The team identified 3 main types based on where the proteins end up inside cells. Cytosolic abundant heat-soluble proteins, known as CAS, float freely in the main body of the cell. Mitochondrial abundant heat-soluble proteins, or MAs, protect the energy-producing mitochondria. Secretory abundant heat-soluble proteins, called SAS, get sent outside the cell. But there is more. Some tardigrades use an alternative pathway involving 2 additional protein families. These were first discovered in a species called Echinuscus testudo, so researchers named them Etas alpha and Etas beta to understand how these genes evolved. The research team looked across 13 different tardigrade genera. They found 74 CAS sequences, 8 MAs, 29 SAS, 22 Etas alpha, 18 Etas beta, and 21 sequences of a broader stress resistance gene called MRE11. This allowed them to build the very first evolutionary trees for these gene families. What they found was surprising. Instead of a simple story where genes duplicated once and spread, they discovered many independent duplications happening across different lineages. The genetic history of anhydrobiosis turned out to be far more complex than anyone expected. One of the most puzzling questions about tardigrades has been how they handle radiation. These tiny creatures can survive roughly 1,000 times more radiation than humans can, which is an astonishing difference. For years, scientists wondered whether tardigrades simply had some kind of shield that prevented radiation from damaging their DNA. In the first place, it seemed like a reasonable explanation for such extreme tolerance, but research from UNC-Chapel Hill, led by Bob Goldstein and published in Current Biology, revealed something far more interesting. Courtney Clark Hatchell, working in Goldstein's lab, discovered that tardigrades are not actually immune to DNA damage at all. When exposed to radiation, their DNA gets damaged, just like ours. The difference lies in what happens next. Tardigrades mount an absolutely massive repair response. They dramatically increase the production of genes involved in DNA repair, pushing these repair proteins to become some of the most abundant molecules in their entire body. It is not that they avoid the damage, they just have an extraordinary ability to fix it. Think about that for a moment. When radiation hits a tardigrade, it breaks apart their DNA, just as it would break apart yours, but then the tardigrade floods its cells with repair machinery, piecing everything back together with remarkable efficiency. It is like having a construction crew that can rebuild an entire house in hours after a devastating storm. Around the same time, researchers in France reached similar conclusions through independent work. Jean-Paul Concordat and Anne de Sion identified a new tardigrade protein that appears to protect DNA, adding another piece to this puzzle. The combined findings from both teams suggest that tardigrades have evolved multiple strategies for dealing with genetic damage. Their approach is not passive protection, but active aggressive repair. This makes their survival skills even more impressive than scientists had initially imagined. The story of tardigrades in space research begins back in 1964, when scientists first proposed sending these remarkable creatures beyond Earth's atmosphere. Their reasoning was straightforward: if tardigrades could handle extraordinary amounts of radiation here on the ground, perhaps they could teach us something about survival in the harsh environment of space. But it would take decades before anyone actually put that idea to the test. The real breakthrough came in 2007 with the Photon M3 mission. European researchers loaded tardigrades onto an unmanned spacecraft and sent them into orbit for 10 full days. These tiny animals were exposed directly to the vacuum of space, completely unprotected, when the spacecraft returned to Earth, and scientists rehydrated the specimens. Something astonishing happened. The tardigrades simply woke up and carried on as if nothing had happened. They had survived not just the vacuum, but also the intense ultraviolet radiation from the sun. This success opened the door to more ambitious experiments. In 2011, the Space Shuttle Endeavour carried tardigrades to the International Space Station as part of mission STS-134. This allowed researchers to study them in more controlled conditions over extended periods. Then came an unexpected chapter in 2019, when the Israeli lunar lander Bereshit crashed on the Moon's surface. It turned out to be carrying tardigrades among its payload. Whether any survived the impact remains unknown, but in a strange way, they may now be the first Earth animals to have reached the lunar surface as permanent residents. What makes tardigrades so valuable for this kind of research is the combination of their extraordinary tolerances and their practical advantages. They can handle freezing temperatures, high radiation doses, and near-complete desiccation all at once. But unlike many extremophiles, tardigrades are also relatively complex animals with distinct body structures and behaviors that scientists can easily observe and study. This makes them an ideal model organism for astrobiology, offering insights into what life might need to survive on other worlds. When James Fleming, David Pisani, and Kazuharu Arakawa set out to trace the evolutionary history of tardigrade survival genes, they had a pretty straightforward hunch. They assumed that gene duplications would line up neatly with habitat transitions. The idea was simple enough: as tardigrades moved from aquatic environments onto land, they would have needed new genetic tools to handle drying out, so the researchers expected to find clear evidence of ancient gene duplications that corresponded to this ecological shift. It seemed like an intuitive way to connect genetic changes to real-world adaptations. But what they actually found was far messier and more interesting. The team analyzed sequences from 6 different gene families across 13 tardigrade genera, including representatives from both major lineages, the eutardigrades and heterotardigrades. They identified dozens of heat-soluble protein sequences and constructed the first phylogenies for these gene families. What emerged from this analysis was unexpected. Instead of a few ancient duplications that could be easily tied to habitat changes, they discovered numerous independent duplications scattered throughout tardigrade evolutionary history. This finding painted a much more intricate picture of how anhydrobiosis-related genes evolved. The evolutionary history of these desiccation genes does contain remnants of ecological history, but it refuses to yield any simple explanations. There was no clear link between species with strong anhydrobiotic abilities and particular gene duplication events, the genetic landscape underlying modern tardigrade extremitolerance turns out to be surprisingly complex, with different lineages apparently developing their own solutions to the problem of survival without water. This complexity suggests that tardigrade resilience is not the result of a single evolutionary innovation, but rather a patchwork of genetic changes accumulated over millions of years of adaptation.